Alrighty, so my name is Kirk Butler, and I'm a PhD student in Tracy Cookbay's lab at Binghamton University in upstate New York. We're a tissue engineering lab, and we generally focus on heart cells, which is what I'll be talking about this afternoon. Much of our work is based on pluripotent cells, which, as you've just heard at length, have the ability to differentiate and become almost any kind of cell you can find in the body. That includes heart cells, and we're using iPSCs to study cardio cells, or EPICs, and cardiomyocytes, which you'll see shortened to CM throughout this presentation. So, what are these cells? Both EPICs and cardiomyocytes are components of the heart wall. I would imagine this cross-section is familiar to most people in this audience, but in brief, the myocardium is the muscle layer that contracts and pumps blood, and it's sandwiched between an inner endothelial layer that insulates it from the blood and an outer epithelial layer called the epicardium. My work is focused mainly on this layer. So, where does it come from, and what does it do? The epicardium forms pretty early in development, about 21 days after fertilization. At this point, a population of cells from the proepicardial organ migrate onto the myocardium and envelop it. These cells are very important. They produce a lot of the heart's ECM structure, notably fibronectin, and they're also a major source of cardiac fibroblasts. Something like 80% of cardiac fibroblasts are epicardium derived, and it's largely that population that mobilizes to generate scar tissue during wound repair. What I'd like you to keep in mind is that epicardial cells are migratory and they undergo EMT regulated by TGFB. To broaden the scope a little, my work takes place in a larger context of cardiac tissue engineering, where we're attempting to combine different heart cell types in vitro. EPICs I've already talked about, but smooth muscle cells, fibroblasts, endothelial cells, adipocytes, all of these are important, and we expect that combining them will have a beneficial effect on cardiomyocyte function, since you'd be getting closer and closer to recapitulating the cellular landscape of a real heart. Now, on the engineering side, there's a variety of ways you can actually do that. Cell and scaffold type structures that can be used to build grafts, cardiomyocyte rings, we have some other people in our lab working on that kind of architecture, and spherical microtissues. I'm using that type of engineered tissue to model the migratory behavior of epicardial cells and explore how they interact and self-organize in the presence of cardiomyocytes. We think that putting these cells together will improve the cardiomyocytes functionally. So, let's get started. We're going to begin with differentiation from iPSC, and then we'll move on to formation of our 3D microtissues and migration of EPICs into 2D. We start with iPSCs, and we differentiate them by modulation of the Wnt pathway. Apologies in advance for the deja vu here. We start by activating Wnt, and then knocking it down, and if you add insulin after that, you get functional cardiomyocytes by day 9 or so. Aside from the fun way, we can identify these cells by expression of cardiac troponin T. This is a sarcomere component that's heart specific. Now, if instead you pick the cells up at day 6 and replate them on gelatin, then by day 11 you get this population. These cells have an epithelial morphology, they're often multinucleated, and they express the transcription factor WT1, which is characteristic of epicardial cells. So let's look at these populations. We confirmed our phenotypes using flow cytometry and found that by day 11 we lose pluripotency, and the EPICs are typically about 80% positive for WT1, indicated in light green. When we compare them to cardiomyocytes, we find those cells are typically 60% positive for troponin T, indicated in pink. We've also found that each population contains a substantial number of cells that express the opposite marker. About 20% of cells in the EPIC group express troponin T, and about 5% of the cells in our cardiomyocyte group express WT1. So they differ in proportion, and I think a good deal of what the EPIC diff does incidentally is to filter out cells that don't like the replate. Once we have these cells, we singularize them, and then seed 10,000 of them into each well of an ultra-low attachment U bottom plate. We do this for both cell types separately, so I end up with one plate of EPICs and another plate of cardiomyocytes. Once they're seeded, we spin them down briefly, and then after 24 hours, we get spherical microtissues that look like this. You'll see that shortened to MT. They're generally about 300 to 500 microns in diameter. Our EPIC microtissues are generally a little smaller. Their average is 400 microns. We can actually make these microtissues using two different populations of EPICs. So far, and spoiler alert from now on, we've been looking at microtissues made from day 11 EPICs. But if you plate them back down at day 11, you can culture EPICs up to confluence again and passage them. We call that P1. And if you're interested in what happens over subsequent passages of epicardial cells, you should definitely talk to my buddy Saif, who is also here and has done a lot of work with higher passage EPICs. Anyway, I have found that day 11 EPICs are better for forming microtissues. Anecdotally, they form more reliably. I've had multiple P1 seedings fail. And when they do form, they start out very loosely held together and then compact down a lot. Day 11 microtissues remain consistent in size. When we performed immunohistochemistry on these tissues, we found that in the day 11 group, the WT1 positive cells were localized mostly on the surface of the tissue, and in both cases we observed large acellular spaces. More on that later. So, to study the migratory behavior of epicardial cells, I devised this experimental setup. I wanted to know how these cells would migrate as a population, rather than the random clusters you get when culturing them normally in 2D. My solution was to transfer epic microtissues 24 hours after formation into a flat bottom well. Then that allows the cells to attach to the surface and migrate away from a fixed point over 3 days. I'm taking inspiration from explant studies here. What we found is that the epicardial cells move outward from the microtissue and form a skirt region, which we used cytoskeletal immunofluorescence to measure. In order to understand what this population looks like, my mentee Justin and I wrote some MATLAB code which segments an image like this one based on tight junctions and computes the properties of each cell. When we looked at that population, we noticed that the cells nearer to the skirt edge, indicated by a star here, seemed larger. To see if that's real, I binned the cells based on whether they were closer to the edge, calling those leaders, or to the microtissue, calling those followers. We looked for a difference in area, and sure enough, the leaders are 1.7 times bigger than the followers. We've chosen that terminology following the work of the postgroup in zebrafish. They found that danio-epics are divided along similar lines as they migrate, and the bigger cells form a wavefront that leads the migration. This is the first time that I'm aware of that behavior exists in human cells, though it has been occurring in mouse before. I was also interested in how epics would respond to various extracellular matrix coatings. We segmented microtissues expanded over various surface types, and our results indicate that the growth factor-reduced matrigel provides the most favorable substrate for epic migration, improving on 0.1% gelatin by a factor of 1.4. The other surface treatments didn't improve on gelatin. I note that while matrigel is the best substrate for epic migration, it's also expensive and proprietary, and the cells do grow just fine on a gelatin, so that should be okay for this purposes. Okay, so switching gears. Let's talk about what happens when epics and cardiomyocytes are co-cultured in 3D and how epics migrate in this context. What I'm doing here has a lot of moving parts, so I'm going to start with the schematic of the experiment. Up to day 11, it's mostly the same as before, but there's a few things to point out. First, at day 10, I impregnated the epics with cell tracker red. This is a red fluorescent tag that allows me to tell the difference between the cell types when I bring them together. Epics will have cell tracker, cardiomyocytes will not. The other important thing to note on this slide is the timeline after day 11. I see the microtissues here, and then after 24 hours, I transfer the epic microtissue into a well that already has a cardiomyocyte microtissue, as shown in this diagram. Once they start to interact, we have time points at day 1 and day 7, shown here, where we take bright field and red fluorescence images of each microtissue, and at day 7, we record 20 seconds of GCAM calcium imaging at 100 frames per second. I'll talk more about that in just a minute. Here's what this process looks like visually. We start with pluripotent cells at day 0. By day 11, they're epics, and we seed them into microtissues and split them down. Then we perform the transfer. After 7 days, the two tissues merge together. Using a combination of cell tracker and the GCAM signal, we can locate the epics and cardiomyocytes within the structure, respectively. We find that the microtissues compartmentalize with cardiomyocyte and epic-rich sections. With image analysis in MATLAB, I'm able to calculate how the cardiomyocyte region changes over the course of the interaction. And we find that it increases in cross-sectional area by a factor of 1.5. In the size analysis, I segmented the bright field image of each microtissue to identify the cardiomyocyte-rich compartment. Using that segmentation, I was able to measure how much the cell tracker tag group overlapped it. What I'm measuring here is where the cell tracker image on the left coincides with the dark blue compartment on the right. We found that by day 7, 39% of the cross-section had been infiltrated by epics. I mentioned at the start of this talk that epics can undergo EMT, and that process is regulated by TGF-beta. We typically culture our epics in the presence of TGF-beta inhibitor to prevent spontaneous EMT and loss of phenotype. But I wondered what would happen if you remove that inhibition from the merging microtissues. The result is a pretty striking difference in morphology, with the inhibited tissues developing many of the large acellular structures I talked about earlier. I haven't been able to run histology on the merged tissues yet, but I think those gaps are what you're seeing in the lighter regions there. In general, we found that the uninhibited tissues were more compact and smaller by day 7. We obtained functional information from GCAM. All the cells in this experiment express this modified calcium channel that flashes green fluorescence under calcium flux. We've used this to measure the contraction characteristics in ImageJ using the muscle motion plugin. I'm showing a typical contraction profile here. Also, motion gives us information about the shape of the contraction profile. For these data, we're looking at contraction duration and peak-to-peak time, as well as time to peak, which is from the start of the contraction to its maximum signal, and relaxation time, maximum signal back down to 0. I've indicated visually on this example profile what we're actually measuring here. We found that for all of these metrics, the merged microtissues show a decrease compared to microtissues made from only cardiomyocytes. This reflects a contraction that's shorter, sharper, and more rapid. We interpret this result as suggesting that the presence of EPICs as a supporting population has a positive impact on the contraction mechanics of the cardiomyocytes. I will note that that's not a particularly surprising result, since there's already extensive research that shows that cardiac fibroblasts have a similar beneficial impact. Bringing it all back together, we found that human EPICs can be used to construct 3D microtissues, and they remain migratory in that context. We found that day 11 EPICs are better than peak 1 if you're going to do this. When we examined their migration in 2D, we found that they migrate in a wavefront that's consistent with behaviors found in animal models, and that matrigel is the best substrate for EPIC migration. In our 3D studies, we found that EPICs will migrate and interact with cardiomyocytes, similar to the interaction of the protocardial organ and the formation of the epicardium in vivo. And we noted that without TGF-beta inhibition, we get more compact structures, possibly indicating more robust migration. Finally, based on our calcium handling results, we infer a positive functional relationship in 3D co-culture. In continuing studies, we're planning to explore EPIC migration in 2D more closely, hoping to see the leader-follower transition in real time. We'll also be performing more co-cultures both in 3D and both in 3D as I showed here, and in a 2.5D format. And we're planning to expand on removal of TGF-beta inhibition to more direct control over transdifferentiation of EPICs. The figure shown here is a pilot study for that. In conclusion, I'd like to thank my advisor, Tracy Cookway, and the rest of our lab, in particular Natalie Packer and Justin Jablonski. And I especially want to shout out Saif Ahmed, who contributed a lot to this project, and you should definitely check out his poster in the session right after this, number 269. Thanks for listening.
Wonderful talk. Any questions?
Yes.
Great talk, and I'm very interested in your epicardial kind of the leader and the follower. It seems in your experiment there are lots of heterogeneity of the epicardial cells using the protocol that we developed.
Mm-hmm.
And I'm wondering, do you know any kind of molecular marker for the between the difference in the leader and the follower cells?
I'm actually very curious whether there is a particular marker that would differentiate the two. This is pretty new work in animal models too, and I don't think the postgroup has identified a particular marker that would identify these cells that we could look at. I think one potential avenue for this is in order for the cells to adopt that larger morphology, the way that it works in zebrafish anyway is they generate a lot of extra genetic material by endocycling, and over the course of that process they get larger and often generate multiple nuclei. So I think you could potentially look using something like Fuji for like cell cycle activity. But as far as the actual leader-follower differentiation, I'm not sure if there is any particular marker that's known at this moment.
Okay, so you haven't done any like anesthetic or bulk or single cell anesthetic to see the difference?
Not yet.
Oh.
Any other questions? Well, if not, let's thank our speaker again.